is phenotyping. We want to use digital tools to assess patients, quantitatively track them. The middle layer is molecular. We've developed molecular blood tests, liquid biopsies for different mental health indications. And the inner layer is therapeutics, bringing people to the right treatment and repurposed drugs. And these are very common disorders. All of us have patients or family members or friends who have suffered from them. And this is the example, the tragic example of the sister of one of our investors who was an executive in Silicon Valley, had access to the best possible care, was misdiagnosed with depression, was treated with a series of antidepressants, ended up committing suicide. So even with the best sort of care that money can buy currently, there are tragic outcomes, not to speak in sort of the larger mental health population with severe mental illness, veterans, and so on, where we're losing 20 veterans every day. So we need to do better, all of us as a field, and that's what we're trying to do with our approach. So this is a little bit the science behind it. We started on this journey over 2 decades ago. Can all of you see this properly and hear it? Is it fine? Okay,
yeah.
Good.
But we can't see you though. Maybe you have to.
That's okay. That's okay. I'm not that important for the purposes of this, but I will after that at the Q&A I'll jump in.
Okay.
So so this was initially a dream about 20 years ago when I started my first lab at UC San Diego while I was still a resident. I was coming into the psychiatry residency training after having done a PhD in cancer molecular genetics at Scripps and a postdoctoral training in neurobiology at Scripps with Gerald Edelman. So I had that, background then, sort of all that excitement that was occurring in the cancer field, where you were doing genomics, molecular profiling, the first Affymetrix array were coming on board. It was a very exciting time for cancer diagnostics. And when I moved to psychiatry, the sort of new and virgin arena, the goal was sooner or later to bring some of those approaches to bear upon mental health. Why not have sort of similar precision in these very common and very disabling disorders? So that was the dream. And then we've done 20 years of science, and now some of that science is in products that are available to people. So our work was very translational, and by translational, it means that we've done a lot of human studies, animal model studies, integrated that to, there are things you can do in people that you cannot do in animal models, relevance to the disorder and so on, specificity, and there are things that you can do in animal models that you cannot do in people, obviously. A lot of direct brain studies, brain-blood interactions, and so on, trying different compounds. And when we first started on this journey, this was my sort of simple-minded view of psychiatry. So that's, psychiatry for dummies, and in that category, I include myself. It was a very simple view that probably we have 3 broad domains: the anxiety domain, the mood domain, and the cognitive domains. These domains are overlapping, and that stress is a major trigger of psychiatric disorder. So that was the simple sort of view that we put forward. And then over the last 20 years, we've mapped the territory. These are all studies from my group, genetic and genomic studies, mapping the territory in terms of the genomic landscape of mood disorders, cognitive disorders, anxiety disorders, stress, alcoholism, which overlaps with the other major domains. And there were some interesting offshoots from these works. So from the cognitive domain, we branched into Alzheimer's disorder. From the mood disorder domain, we branch into studying suicide, which is an unfortunate negative outcome of mood disorders, but not just mood disorders, the final common pathway of a lot of these psychiatric disorders when they're not treated well. And then there was some serendipity involved, and we found that some of the same pathways and molecular players that were involved in mood disorders, anxiety, and stress also are involved on the flip side on longevity. So there is sort of light at the end of the tunnel. Not only we hope to help people treat their mental health, but we could actually make them, as a result of that, have a longer lifespan, longer health span, which is something that's of active interest to all of us. So anyway, the big bet that we made in 2004 when I was recruited away from UC San Diego to Indiana University Medical School was that we should have a mini Framingham type study for mental health disorders, a population that we follow longitudinally. We can look at people, over time, what changes in their blood, what changes, in their behavior, what are the clinical outcomes. And these types of longitudinal approaches are extremely hard to do. Obviously, they're time consuming, so we've been at it for 17 years now, but extremely powerful because if you catch people in different states when they're not sick, when they're sick, and so on, and you look inside the same person, what changes, you have with what's called technically this within subject design, a lot of power to detect signal, orders of magnitude more power than the current sort of large-scale genetic studies that are being done, which are sort of cross-sectional case control. So that's sort of all technical terms, but this was a big bet that we made. The second big bet was to go for gene expression as opposed to DNA. Genes and environment interact at the level of gene expression, RNAs, and that's where the action occurs. That's where the disease manifests. It's dynamic and so on. And of the RNA, you have the proteins being made that underlie the biology. So we went for gene expression, which again is orders of magnitude more powerful than just looking at DNA. On all these people, majority of them, we had electronic medical records, so we can look at outcomes, blood samples, or testing that we did years ago. What was the clinical outcome, which helped us with our studies. And in addition to this, the third thing that we did, we were very programmatic about understanding disease in a quantitative fashion, what we call phenomics. So we developed some of our own questionnaires that eventually are now into phone apps, and we were very systematic about using targeted quantitative phenotypes for discovering our biomarkers, not the broad diagnostic classification that existed and still exists in psychiatry. So those were the innovations, learn from cancer. We had a convergent approach that we pioneered in terms of identifying and prioritizing genes and biomarkers. Phenomics is the attention to phenotype, being very translational about things, being very systematic, the 4 steps that we take in terms of biomarker discovery, prioritization, validation, and testing, and then these 3 concentric circles going from phenotype to molecular markers to treatment. So those were the ideas that drove all this work. And when we started it, cancer was 15 years ahead of psychiatry. I think now with the work that we've done and others in the field have done, we're catching up, and we may be able to, in the next 3 to 5 years, to be on par with cancer in terms of some of the things that are being done. Now, we still have, a steeper climb as a field, because in cancer you can biopsy the tumor, and you can have much more accessibility to the pathology. In psychiatry, obviously, you can biopsy the brain in live individuals. So that's why we went for these peripheral markers, which were not a given that they could have been identified when we first started this work. Now, both in cancer and in the work that we're doing, we are relying on this liquid biopsy approach. So cancer is not just relying on biopsying tumors, but now they're developing this blood test, so-called liquid biopsies. In our case, biopsying the organ wasn't an option, so we went from the beginning for the liquid biopsy approach. And we want to get a cover like this as a field in a few years, where it says psychiatric treatment gets personal. In fact, in some pockets of the country, of the world, in some pockets of what we do in our group and with the company, this is a reality. It's like that William Gibson quote, "The future is already here, it's just not evenly distributed." And it's the mission of MindX to distribute it more widely and more evenly. On the phenotypic side, one of the big discussions in psychiatry is, do you put people into boxes, diagnostic labels, or should you take a dimensional approach? And we took a dimensional approach in our work and in our thinking from the beginning. And now with the tools that we have at MindX, we can quantify through our apps or through our blood tests people's levels of anxiety, their mood, their cognition, and really have sort of quantitative data for having people in this multidimensional landscape. And that's probably a better description of an individual than putting sort of DSM labels on them. So at some point in the near future, some of these things will start to appear in the apps that we have and in the reports that we give to doctors and to pharma companies. And, that's we're going from the old to the new. Right now, psychiatry seems puzzling. There are many things that we need to make sense of, but we think that with these dimensional approaches and with quantitative and approaches and with precision, we can, be ahead and be part of the 21st century, not part of the 19th or early 20th century. So the blood biomarkers, it was really an issue of looking for the keys under the lamppost, because a priori there wasn't, when we first did those studies in the early 2000s, mid 2000s, when we started publishing them, it wasn't clear that you would find something in the blood that would track CNS pathology, that would track brain disorders. But we went for it because we thought, if we manage to find something, then it's something that can be deployed widely in clinic and so on. Brain imaging is not something that's widely scalable, at least wasn't then, maybe with the technology of the future. EEG seemed very complex and sort of, black box to us. And molecular markers are, may give you a window into the biology, may match people with medication. So we went for that. And there are three reasons really why you can pick up some signal in the periphery, although you have to be very careful to identify that signal and validate it. So one of it is that the nervous and immune system have common developmental roots. They're the most complex and interesting sort of tissues that we have, and they have a lot of interactions. There are these brain-blood interactions, the immune system can influence the brain, and so on. And in the end, you have this sort of similar gene expression signatures in different tissues, in this case, brain and blood, that are due to some of the same underlying genetic architecture in these tissues and some of the same external factors that affect these tissues, whether it's stress, hormones, medications, and so on. So for those reasons, with care, you can pick a signature. It's sort of finding the needle in the haystack type of thing. There are thousands of things that change in the blood. A very small proportion of them are relevant or track the brain phenotypes, the mental health phenotypes that you're interested in. But if you do find them, as we did, then you have a window into understanding the disease. You have some help with making an assessment, and you have some help in terms of matching people to the right treatments. So, when you make big claims, one of my friends, Danny Weinberg, and I think he's quoting somebody else also, when, big claims require extraordinary proof. Right, so when you want to claim that you found something in the blood that's a biomarker for a complex disorder like one of our psychiatric disorders, you have to be very careful that indeed you did find something, and that you're not fooling yourself, fooling the field, and giving false hope to patients. So we developed this very systematic approach to discover biomarkers. There are 4 steps. There's a discovery step that's based on a longitudinal design, following people over time and seeing what changes from visit to visit in their blood. That's a very powerful approach. I won't go into the technical aspects of it, but for example, a colleague of ours in not in psychiatry, but in sort of genomics, Mike Snyder at Stanford, also independently demonstrated the power of these approaches, these longitudinal approaches. He published a beautiful paper in 2012, 2013, I think, in Cell with an N of 1. The study had an N of 1. It was himself, followed over time, over a year, and he showed the power of actually tracking within a person markers over time, catching early some signs of diabetes, even though he was a avid cyclist and other things. So our cohorts have dozens of mini Mike Snyders in them for each disorder, and that's how we initially discover candidate biomarkers. Then we go to the prioritization step, which is a very bioinformatic heavy approach where we look for convergent evidence. We have large databases where we've curated thousands of papers from the field, so any signal that we get from our discovery studies cross-validated against any other data that was ever published in the field, and that's all sort of internal databases that we've developed over many, many years. The third step is validation. So once you have markers that candidate biomarkers you discovered, you prioritize, then we go into an independent cohort of people who are really sick to see if there are change even more there. And in the case of suicide, for example, those were suicide completers. So we had blood samples from the coroner's office and we validated our markers that way. In other disorders, these are people who are clinically severely sick by clinical measures, questionnaires, hospitalized, and so on. And then after the first 3 steps, we have a list of the biomarkers that make those steps, that have the most evidence, and that list is then taken into testing for clinical utility in additional independent cohorts where we see, can we predict somebody's state, how sick they are at that moment in time? Can we predict their immediate risk, whether they'll have any hospitalization or ER visits in the 1 year following testing? Can we predict their long-term risk, if they'll have any future hospitalization or ER visits or suicides? So that's how we come up with our biomarkers. In the end, we end up with a very small panel. These are typically for each of our indications, 12 to 18 markers that we can provide, based on an algorithm, reports to doctors that I'll show briefly in this. So, what's the accuracy of this test? Well, it's when we test our different panels in all comers, it's slightly above 70%. It's in many cases better than that, but we always like to sort of quote the lowest numbers. So it's above it's a little bit over 70% in all comers where we test our panels. And these are all numbers from independent cohorts from population or patients that were not used in any way, shape, or fashion for the discovery of the biomarkers. So about 70% accurate in everybody. We also have developed digital tests, which are over 80% accurate in all comers. So then one might ask the question, why use an expensive molecular test when you can actually get the better classification and prediction with phenotypic tests, which is a very good question that we also asked ourselves. And the answer to that is twofold. First of all, with the molecular test, you can also pair people to medications, to suggested medications based on biology, like Foundation Medicine did and other companies did for cancer. So that's very important. You can't do that readily with phenotypic tests. The second thing is that the two are not mutually exclusive. They're synergistic. So when you pair, if you have in somebody their molecular testing with their digital testing, then you actually go to over 90% accuracy. And that's how we do it with our algorithms. Moreover, in certain high-risk groups, just the molecular tests are over 90% accurate, such as tests for suicide risk in males with bipolar disorder or males with depression. And then in the high-risk groups, the algorithm combining the genomic and phenomic data is very accurate. And those numbers are then clinically actionable and comparable to other things in clinical medicine. So, and I'm illustrating here with a few examples. So our work on suicide, that's the algorithm combining the some phenotypic measures that are now in an app that we have there in the Apple and Google store, called the Life X Mind app, Life and Mind app by MindX Sciences, combined with the suicide risk digital score, the SX Prevent, combined with the molecular test score. You get over 90% accuracy across all comers and in different subtypes of suicidality that we identified and in different diagnostic groups. And those little stars over there are, you know, statistical significance. 1 is nominally significant, which is okay, and 2 is survives Bonferroni correction, which is a very high stringent level of statistical significance. So these, you know, are now in the range of other things in medicine, and we think they are, you know, usable and a good starting point. The alternative is what? 0 predictive accuracy, because you don't have any other tests out there to help you with your clinical assessment. So then our initial work was, you know, well received when we started it. This is a funny story that I often tell, you know, tongue-in-cheek. We submitted our initial study on, for example, for the suicide biomarkers in 2013 to Nature and Science, which are 2 sort of you know, tabloid journals in science, very sort of red journals, and both of them rejected it. They didn't even send it out for scientific peer review. They just weren't interested in the paper. So then we published it in a good journal, but not Nature and Science. And then after that, they both wrote 2-3 page pieces about it, in their news and views sections and comment sections. So, you know, in some ways, you know, academia is a sort of a juvenile game, and we feel sort of that, you know, being in the real world and trying to help doctors and patients is where the future is. So, well, you know, we've published additional studies since. We've looked at the reproducibility of some of the things that people are putting out there from genetic studies called GWASs with our biomarkers, and a lot of the GWASs actually converge on our biomarkers. They don't reproduce each other because they look at DNA mutations, but at the gene level, they do converge on our biomarkers, which is, you know, how the biology works. So there's a lot of reproducibility. And, you know, our work was also described in New England Journal of Medicine Journal Watch, which we were happy with that because that's, you know, a place where a lot of primary care doctors and other doctors look. We've done similar studies on pain, which is another sort of tough nut to crack, right? So as a physician, you have to rely on what the patient is telling you in terms of their pain, what you know, your clinical impression is. So we found biomarkers for pain. And again, I remember sort of talking to a colleague from Stanford, who I won't name his name, but he's a big name in the pain field. And about 4 or 5 years ago, which was a year before we started doing the studies, and saying, well, you know, we're doing all this stuff in psychiatry. Should we, you know, pain is very similar. It's subjective. Why don't we go for some biomarkers for pain? And he gave us some good advice. He also said it's not going to be possible to find biomarkers in the blood that are reproducible. So instead of discouraging us, that motivate us, we went for it, and we were lucky. We did find biomarkers that are reproducible, and we did find, you know, predictive and so on. And in the end, I think he was one of our reviewers on our study, and he was very fair, and our paper got published, and then we got a lot of sort of follow-up visibility and work on it. But you know, don't always let you know, experts discourage you. If we had been expert in anything that we did, we would have never done it because it's very tough. So a little bit of ignorance and enthusiasm goes a long way. So anyway, that was the pain work. The important thing for all these blood tests is the pharmacogenomic part, where you match people to medications based on which biomarkers, and all these sort of like names that you see here are names of biomarkers, genes. We're looking at an RNA level, obviously. So you know, if your patient has this and that biomarker, then you know they should be on a for example, in pain, on an antidepressant or on an anticonvulsant or on other. And the name of the game here is, matching people with non-opioid medications, because we all saw what problems opioids created for us, right? In addition, you can use these biomarker signatures to find existing drugs used for other indications, repurposed drugs. So that's an area where MindX has a strong in-house effort now. And we've done similar work recently for mood, which came out a couple of weeks ago. We've found a panel that not only can provide objective information on depression when somebody's in a depressive state, but also of their bipolar risk, matching with medications. So you can generate a report for doctors. This is sort of from the academic publication. The report that we have at the company is in a much nicer format, but in essence, the information is similar. You're providing a score for severity, future risk, list of medication. So in many ways, it's like a credit report, like a FICO score for the disorder that you're studying, where you have a number and then the list of things you can do to mitigate the risk, which in this case is medications and things like that. So those are the three circles that went into the company. There are digital phenomic work, our molecular genomic work, and our repurposed drug therapeutics. So that's the story behind this in a sort of very high level view fashion. At the company side, we're strongly mission-driven. These are very common disorders, as I mentioned, and I'm a psychiatrist myself, clinical psychiatrist, in addition to being a geneticist, researcher, etc. So I still see patients a day a week, and so when I say 19th century medicine, it is what it is. I mean, we have a few objective tools now, but pretty much we're doing it like our colleagues were doing it, over 100 years ago or more. Not that they were doing a bad job. I mean, Kraepelin, Euler, Freud even had interesting insights, but we need to do better, okay? If Freud was alive today, he would probably be a molecular psychiatrist. He was very innovative for his time. Anyway, how we're doing it at the company is we have this platform approach from digital to molecular to therapeutic and a pipeline. And that's the team that we've put together. So besides myself, we have Sunil Hazarai, who on this Zoom actually appears as Chris Wright. Chris Wright is our CFO who set up the Zoom call, but was not with us there. But that's actually Sunil Hazarai, our president. Strong executive leadership at various diagnostic company, Roche Diagnostic, other, and on the medical side, the interaction with pharma, we have Steve Stahl, who's one of the better known psychiatrists in the world, has worked in industry, academia, authored textbooks, was my mentor when I was a resident at UCSD. Then in terms of our efforts, we have Felix leading our digital efforts, Sunil Kurian at Scripps, our molecular efforts, and we have a new guy, so that's why he's still a square. We have, how you fit a square peg in a round hole. An executive in residence who's looking at the drug repurposing part of things. And, this is all sort of like on the company side, but we have, a board, we have investors, we have a scientific advisory board, and these are all interesting people who have a strong track record, who have a passion for this, and bring a lot of experience to doing this type of work. So this is our pipeline. We have 6 reports that we can provide now in an early access program to doctors for mood disorders, for suicide risk, for PTSD, for pain, for longevity, and for memory, Alzheimer. So those 6 are readily available. On all of these, we've published papers, although the products of the company are a bit more advanced and beyond what we put in our publications. And we have other indications that we are hoping to roll out first with publications later this year. The studies are done. We're just sort of in various stages of submission and peer review. So what we have ready to go right now are 4 products. We have the Life and Mind app, which is in the Apple and Google store. This is sort of a Fitbit for the mind. You can track your mood, anxiety, stress, pain, all the phenotypes for which you have blood tests. We have, we have the phenomic part of them in the app, the digital assessment. It's a very cool app. It's in essence a medical device masquerading as a consumer product. So it's out there in the store. Anybody can get it, use it. It's a portal for people to, it's its own freestanding useful thing, because why just sort of get assessed once every 3 months or 6 months when you see your doctor or therapist? This way you can keep track at home, have quantitative curves to share with your doctor, with your therapist, and so on. So it's its own nice freestanding product. These are all questionnaires that we developed over the last 15 years, validated against the current ones used, and were the ones, our questionnaire were the ones that we use for our biomarker discovery, because they're more quantitative, more cleaner, more sort of phenotypically relevant to the disorder. So now they're all in a nice usable format. So that's the LXM, the Life and Mind app. Second product is SX Prevent. This is a suicidality risk score that we developed. Doesn't ask about suicide, so it integrates a lot of other interesting risk factors. For those of you who have read up on genetics or are familiar with that, there's that whole polygenic risk score type of thing in genetic. This is like a polyphenic risk score. You just quantitate 0, 1, 0, 1 different risk factors, and you get the risk score. Obviously, we at the company side, we do it a bit more algorithmically. It's more complex than that, but that's the basic idea. You get the risk score based on all the risk factors that you have and that are relevant to suicide. And even myself, after seeing these types of complex patients for 20 years, I can't juggle in my head 22 risk factors or 30 risk factors or 38 risk factors as some of our more advanced versions have. But this one, quantitates them, provides a score, like a traffic light, high risk, medium risk, low risk. Also provides pivots immediately from assessment to treatment, provides a personalized treatment plan for that person, for their physician to use with them to mitigate risk. And doesn't ask about suicidal ideation, which is something that's, avoided or not done in a lot of healthcare settings. So it can be used widely, online, primary care, anywhere. Then we have the MindX blood test. This is the liquid biopsy. We do all the genes in the genome. We look at their RNA gene expression. We have a large laboratory company that we've partnered with that does these tests for us. We get the raw data, analyze it, and provide reports to doctors. And all this is integrated in our fourth product, which are the MindX dashboards, where physicians or pharma companies or hospitals or insurance companies can use our products, integrate them, and keep track of their patients. All of our products can help clinical practice or clinical trials by matching the right patients to the right treatments and monitoring response to treatment in a quantitative way. So that's the MindX liquid biopsy blood test. It provides molecular testing for multiple mental health disorders. We provide, out of a single test, we can provide different reports a la carte, depending on what the doctor wants. So if you want a report for mood disorders, we can provide that. If you want also a report for pain, we can provide that, etc. We think that there's a lot of value, and we've shown that in our studies on having multiple time points on people. So you worry about one of your patients. What do they actually have? Am I treating them with the right thing? So you do a blood test on them, you get the report, maybe you adjust their treatment, and then you repeat the test a couple of months later to see have things improved. Do we need to adjust course further, and so on. And then when somebody is doing well, they should be part of their annual exam. Primary care doctors get a bunch of blood tubes at every yearly annual exam. This would be an additional extra tube that they get. Primary care doctors are actually much more pragmatic than us in psychiatry. They're busy. They don't think that they are the cat's whiskers and can diagnose people just by looking at them. So they rely on blood tests. They want additional information to help them with their practice. And in fact, they see 80% of mental health patients initially and for a few years are being seen by primary care doctors and internists, not by psychiatrists. So I think that's a population that would be very in fact, we know they're very interested and open to using our tests. What about hospital systems? We are now in advanced talks with the hospital system to use some of our tools. Obviously, it helps them with patient care improvement, and that improves their reputation. Helps them on the revenue side with if they have executive health programs where people can pay out of pocket for these tests, which are not inexpensive. We're working hard to move them down the pipeline towards Medicare reimbursement, insurance reimbursement, but right now it's out of pocket, and that's a barrier for a lot of people. And then they also have employee wellness program, physician wellness programs, and again, offering the latest and greatest may help them with recruiting and retaining staff, especially in this post-COVID world where a lot of hospital systems are losing staff to burnout and other things. So in the end, we have these dashboards that we provide to doctors like you, to consumers. We want to provide them to hospital system and insurance companies and to biopharma, where we can integrate, our digital products, molecular product, and therapeutic recommendations. Now, you don't have to have all 4 of our products. Either one of them might fit your needs or your patient's needs and would work independently of the others. But having this suite of things is optimal, and being part of this ecosystem over time, we can provide you better analytics and information if we have multiple testing visits on you, just like we demonstrated in our studies over the years in people. It's one thing to have a level of a market, it's another thing to have slope over time and other analytics that are longitudinal and informative. Think about, PSA for men among us, right? Prostate specific antigen. If you know you have a certain level, that's one thing. But if you know that the level of your PSA went up dramatically over a short period of time, that's much more informative in terms of your risk. So similar here. And in the end, we're sort of building that dimensional approach of philosophy. We're working on rolling it out in some of our products where people are assessed dimensionally. This is sort of a population of people that we've studied and tested on the research development studies. In red are high-risk suicidal people, in blue lower risk, yellow intermediate, and this was a patient that actually committed suicide, and you can see how she was very strong on an outlier almost on all these measures. She was at the 100th percentile of our biomarker panel and so on. And that's the tragedy when you do research, because you have things buried there in your data sets, de-identified, that you don't communicate to doctors. So this lady, a year after she left our study, we heard that she committed suicide. So then we went into our data, looked at her results, and the risk was there all along and the information. But that being a research study, we didn't have an ability to communicate that to her doctor. So now with the company, we do have an ability. So in the end, sort of the light at the end of the tunnel is that we think that these disorders can be assessed better, treated better, without wasting years of your life. And the flip side of it is that a lot of the same molecular biological pathways, if they are flipped in the other direction, actually lead to longevity and increased health span. So by treating mental health, we are turning on this life switch in the direction of longevity. And that's, an area that, is something that we've done research on. We have a product at the company, the longevity assessment blood test. And I think, in the end, just not just treating clinical disorders, but improving people's lives is what it's all about.